See, will it let me? See if it'll let me open this in Google Docs. Not really. Yeah, I'll just zoom in, I guess. Uh, hmm. Actually, maybe the way to do it is to download it and then
And then share
open
it from your
these.
own. Yeah.
Yeah, let
Percent
me
pip
do that.
install. I didn't know that was a thing. Is that a thing? I have to try it.
Upload complete. That's not the wrong one. Oh, there it is. Okay, good. Yeah, okay. We go in here, and then I'll bring it into a proper viewer. Yeah, here we go. Let's try this. Okay, yeah, so, okay, that's good. Everybody see that? Anybody?
Yep.
I can see it.
Oh, good, thanks. All right, so, yeah, we're on to the next session. So, there's a quick coffee. I should have given myself more time. What I want to do now is spend a little time, not too long, on basic modeling ideas. We won't go into too much depth, but just enough to get you started. This is a PDF that's in the drive. I'm just viewing this on Sumatra PDF, which is a nice PDF viewer. So let's get started. So just a little thing about models. So I mean, modeling in systems biology. I mean, modeling in systems biology became popular after about 2000. Before that, there was some modeling, but not very much. And it was mainly frowned upon. People didn't think modeling was important. It was possible, or what was even the point of doing it? And it was extremely difficult to get support for modeling before 2000. And then what happened was suddenly all this data started to begin getting generated, especially the transcriptomic data at that time. And people wondered, what are we going to do with all this data? And then modeling became more important, and people realized, yeah, we have to do this. And of course, a new generation of molecular biologists and biochemists and so on arose. And today we have modeling is fairly well established in systems biology. And part of the reason was people before 2000. I mean, I did my degree, my undergraduate degree in the 1980s. They had no idea what modeling was. So they didn't even know they were building models themselves, because if you look at any textbook, molecular biology textbook, if you look at that today, you'll see page after page of pictures, cartoons, and every one of those is a model. It's just not a mathematical model. And they never realized that, I don't think. But the minute we put math to it, they were horrified at the idea, partly because, of course, many people then were not trained in mathematics. That didn't help, or trained in any kind of computing, which didn't help either. So, but today now modeling is fairly well established, and modeling, comes in all sorts of shapes and sizes. And the whole point of a model, as I give here in this slide, is to help us understand more clearly the observations we make. That's all they're used for. You have observations, and you'd like to understand why the observations are the way they are, and how can we explain them. And it goes very early on, all the way back to the Babylonians even, people started building models. And because the obvious one was the night sky. In those days, there wasn't any light pollution. And so everybody saw the night sky every night. And if you were in Mesopotamia, the night skies were generally probably clear every night. And so there was a great impetus to try to understand the night sky. And so a lot of models emerged, try to explain the movements of the stars, the moon, the planets, and so on. And that's been going on for the last 4,000 years. Now, since then, of course, we've developed a lot more theories. Now, I should say that the theories that the ancients developed were mathematical. It may surprise a lot of people to know that a lot of the models back then were actually mathematical. Some of them really sophisticated. But there are also non-mathematical models that have risen since. I mean, two from biology is the germ theory of disease. Before the germ theory, people thought disease was caused by vapors, or who knows what. They had no idea what caused disease. But then the germ theory arose, which suggested that disease was actually caused by living agents. Of course, we call those today viruses and bacteria, but it's a purely non-mathematical theory that explained how disease spread, and how you could reduce the chances of getting a disease. The other big one, of course, is the central dogma of molecular biology. That is the idea that all life has this core idea where you have DNA going to mRNA going to protein. Right? That's the central dogma of molecular biology. That's also non-mathematical theory. So theories can be, mathematical or non-mathematical. But in each case, they help us understand what we observe in terms of experimental data. And as I said, models can be all sorts. They can be simply verbal, they can be text, they can be pictures. And molecular biology is full of theories as pictures. Of course, they can be as mathematical equations, and that goes back all the way back at least to the ancient Greeks, if not before. And in modern day, of course, many theory models are also represented as computer algorithms. Okay. So rather than writing down what the model is, we actually encode it as an algorithm. So there are lots of different ways of representing models. So why are models useful? Here I just try to summarize why are models useful. Why not just live with data? What's wrong with data, right? Data on its own is pretty boring, really. It's only when you add theories to it does data become more interesting. But it helps organize our data and, of course, our thoughts. It helps transmit our thoughts to our colleagues. It can be written down and passed from generation to generation. A really powerful aspect of models is they help us predict the future, which humans love to do. They also help us understand how a system operates, which is another thing that humans are particularly like to do. Right? Whatever the evolutionary pressures were as we evolved, one of them was to understand the world and how it worked. And in the modern day, although even way back 2,000 years ago, it helps engineers build new devices, processes, bridges, aeroplanes, whatever. So there are lots and lots of reasons why models are useful. And data on its own is simply not sufficient. Although, with modern AI, maybe we're entering a new era again, where maybe all models will be replaced by AI. I don't know, who knows? That's a very speculative point of view, but who knows?
And
Just to
depressing.
show you, what's that? And
And
depressing, yeah,
depressing, but
yeah, I know, depressing as well.
for other
Well,
modelers, yeah.
there'll be for the models for anybody really. There'll be no fun anymore. Uh, Here are some models as pictures. Here's one of the water cycle. I think it's the water cycle. Well, it's the atmospheric cycle of various things cycling from terrestrial Earth, oceans into the atmosphere. These are very common, these kinds of models, right? See them all the time. But more close to home, here's a classic cartoon from a textbook. Shows enzyme action, okay? Substrate binding to enzyme, enzyme substrate complex, and so on. Those of you who done biochemistry or whatever will be very familiar with this. Okay, so models as pictures, very common. And you can rewrite that model as text too, as well. Of course, you can write it down. All right. So, um, as I said, in when I was an undergraduate, we never did any math at all. I did a biochemistry microbiology degree. We didn't do any math whatsoever. And they just didn't think it was important because they had this and they had this. What's wrong with these, right? Aren't they good enough? Well, as things became more and more complex, that kind of representation, I use pictures and text, just became too difficult to handle. And so we turn to something like mathematics. Now, the reason why we use mathematics is because it's a very precise language. It's a language that's designed for reasoning. Okay. I mean, geometry is the classic example of that, right? Reasoning about 2D and 3D space. It's very well defined in terms of the rules you can use to manipulate. There's no ambiguity. And the nice thing is our computers can be programmed to do math as well. So math is a very useful tool to do all this. Now, it takes some training or education to learn how to use math, of course. But once you know it, it's very useful. Now, there are lots of types of math, though, of course. I mean, there's from the simplest, what I call purely empirical, which is just curve fitting, like fitting a straight line, linear regression, y equals mx plus b, or y equals mx plus c if you're from another part of the world. Purely empirical, which is sort of what modern AI does. But you have other kinds of models, like Boolean models. If you're working with a gene regulatory network, it might be Boolean. We also have automata. If you're working with multicellular models, like a tumor model or a model of a liver or something, you might be using automata. A really common one, of course, and what we will do is differential equations. You might have a purely algebraic model. You might have a stochastic model where there are random elements in the model that you need to represent. We won't be doing stochastic models here, but it's something that Tellurium can do and a lot of modeling packages can do. And last but not least, because we have to mention modern AI, and you probably hear me mention this a number of times, but this is the brave new world. We'll see what it leads to. Okay, so many types of mathematical model. So let's look at a model, shall we? Let's look at our model. So this is what I give to the students in one of my classes. It's a nice simple one because you don't have to know too much physics, chemistry, or biology, but it's a water tank model, okay? What I have here, normally I start off with just a single tank, but here I got two tanks. We'll dive straight into the deep end here, so to speak. Two water tanks. We got the first tank at the top. Water is flowing in from some kind of pipe at a rate Q1, and of course it slowly fills the tank with water. But at the bottom of the tank, there's a pipe that empties the water, and it empties the water at some rate Q2, and that water empties into a second tank, which also slowly fills up. Now, the second tank also has another pipe coming into it, which fills into it, and it also has a pipe at the bottom of the tank, which lets the water out. And our aim is to make quantitative predictions about the heights of water in these tanks. So we have two tanks that have two heights, H1 and H2, okay? Now, to make such predictions, we can't really do that easily with just a picture. I mean, this is a nice picture, right? But I really can't predict what the height of water in tank 2 will be after, say, 20 minutes. I know it'll be higher than 0. That's about it, really. So I can't really do much with the tank. So with just the diagram, so we must convert this picture into a mathematical model. Okay, now in building a mathematical model, we're going to have to make some decisions, right? We're going to have to make some assumptions. And so these are some of the assumptions I'm going to make, all right? So the first thing is I'm going to assume that there's no spillage of the water from the tanks. The amount of water that goes in and out of the tank is conserved. We don't lose water through evaporation. There's no splashing around when water splashes out of the tanks or anything like that. We're going to assume mass conservation, okay? That's not a terrible assumption. That's pretty reasonable. The other thing I'm going to assume, maybe the temperature is constant. Maybe not so important in this case, but we'll assume the temperature is constant. We'll assume the pressure is constant in the atmosphere. Not so important, perhaps, but we'll do that. And we'll assume this Torrielli's law for the flow of water out of the tank. So this is a law that was devised with the 16th century. Can't remember now. but it's an odd law when you look at it to begin with. But the rate at which the water leaves a tank like this is actually proportional to the square root of the height of water in the tank. So you'd think that the rate of Q2, for example, the rate at which water leaves the tank would be simply proportional to the height of water. But it's not, it turns out. It's actually proportional to the square root of the height of the water. So this is something you'd have to work out from, other studies. Now, when it comes to systems biology models, we have our own laws like that. We have Michaelis-Menten laws, we have Hill equations, but you'll have your own laws. In whatever domain you're in, you'll have your own laws about, what describes a process. In this case, water leaving a tank is proportional to the square root of the height. Now, the actual rate at which water leaves will be equal to some constant times the square root of the height of the water. And that's what these K1 and K2s are, okay? So there we got some assumptions we're going to make. I mean, the third one we've got from some kind of measurement somebody made. The first one is a basic physics law. And the second one, maybe not so important. But anyway, these are the assumptions we'll make. Then we have to decide, well, what are the different things in this model that we can use to construct the model, all right? But there are three basic things you can think about. These are first of all, the parameters. The things that we can't really change, but are sort of part of the system that we need to know about. For example, the constant, the K constant. Let me go back to the tank. The K constant, what is that? I mean, that's something to do with the diameter of the pipe, probably. And I can't really change the diameter of the pipe easily. So this is going to be some kind of constant, all right? The other thing I'm going to assume is that the tank is cylindrical, so it has a cross-sectional area, and that'll be a parameter as well. This is not something easily I can change because I'd have to make a new tank with a different cross-sectional area. That's going to be a parameter as well, okay? So there are parameters, right? Now, there are other parameters that are absolutely fixed. For example, force of gravity. I mean, I'm not going to go to Mars or the moon and do this. So I'm really stuck with the force of gravity on Earth. That's a sort of absolute constant that's constant, right? Other parameters are like the temperature, pressure, those are also constant as well. So these are various things that are fixed, okay? Now, the other kind of quantity are these things called state variables. These are the things that are going to change over time, and these are the things that I will be observing, okay? So in terms of the water tank model, it's the heights of the water. I will be observing the height of the waters in both tanks. I'll be watching them change over time. If you're a systems biologist, you'll be looking at the concentrations of things, the concentrations of ATP, the concentration of a protein. These are your state variables. These are the things that you want to predict, okay? And finally, there are going to be some set of inputs to the model. Now, these inputs are slightly different from the parameters. The parameters are effectively fixed. Now, some of them I could, in principle, change, like the diameter of the tank, but I don't want to. Too much work, right? So I'm going to stick those into the parameter list. But there are some things which I can actually change relatively easily, and these are going to be called the inputs. The inputs for the water tank is the amount of water coming in the pipe at Q1 and the amount of water coming into the pipe at Q4. Now, in a systems biology model, the types of inputs might be the amount of enzyme that's being expressed, because these days we can easily change that level, or we can change the amount of if you're doing signaling networks, say in a cancer cell line, you'll have lots of drugs that you can try on the cell line. Those would be your inputs, right? Things you have power to change that could influence the model. So all models pretty much have these three groups, and you have to decide yourself how you're going to divide up the things amongst these three groups. Once you have these three groups, you can then start thinking about devising the model itself. Okay. Now, the key thing in the model are the state variables. These are the things you want to predict. And we are going to decide on building a differential equation model. And those differential equations are going to be related to these state variables. And since we have two state variables, i.e. two tanks, two heights of water, we are going to have two state variables and two differential equations, one for each tank. Okay, now, how am I going to get a differential equation for the height of water? Well, there's going to have to be a little bit of manipulation here, but one thing I do know is that because of the conservation of mass, the volume, the rate at which the volume is changing in the tank is simply the difference between how much water is coming in and how much water is leaving. So I can write down to begin with that the rate of change of the volume of water in tank 1 is the rate of water coming in, Q1, minus the rate of water leaving, Q2. Okay. I can do the same thing for V2. This is the volume of the second tank. This time we have to remember there's a second pipe coming in, Q4, which adds to the water coming in. All right. Now you'll see that when we come to build models of networks, metabolic networks or whatever, you're going to be doing exactly the same thing, accounting for everything that makes something and accounting for everything that consumes something. Okay? Now, there's one complication here. In this case, we want to predict the height, not the volume, okay? So I'm assuming the tank is cylindrical, and therefore, and I know it's cross-sectional area. I know, therefore, the volume of the tank is A times H. I can differentiate this with respect to time to show what the rate of change of volume is, which is the cross-sectional area, which is constant, right, times the rate of change of the height. So immediately I can swap in this for this, okay? Now I can take the area onto the other side, right? So I have this relationship and I have this relationship. I can now swap in this piece into here. And then I can move the area over to the other side and I get my differential equation. I can do the same for V2 and I get that. So that's it, that's our model. All right, we have our model. Two differential equations describing the change in the heights. I need to do something about these Qs. The Q1, I know that's an input. That's the rate at which water is coming into the tank. Q4 is also an input. I know what that is. But Q2 and Q3, I need to change because they're related to the height of the water into the tank. So I need to swap them for that, strange law where the rate of water leaving the tank is proportional to the square root of the height. And I need to swap those in. But once I've done that, I get these final differential equations. This is the final model, okay? Now, what do I do with this? Well, if you're a hardcore mathematician, you'll probably try to solve this analytically. You'll go away for a little while and come back with analytical solutions. For the rest of us, we might not do that. We'll instead get a computer to do it instead, all right? And that's what we will be doing here, of course. Now, most of the time, that's the only choice you have anyway, because these models can get very complicated and there's no way you can solve them anyway analytically. You have to solve them by computer. So how can we solve this by computer? Let's do it the hard way, shall we? So the hard way is to do all this. So I have to write a Python script. So I need to import NumPy. I need to import this funny thing called ODEInt from something called SciPy. Well, I need a plotter and I need a math library because I need the math library because I need to get hold of the square root, okay? Then I need to write a new function, and this function is going to compute these two equations. All right, so I set up the numbers for the constants and things like that. And then I write down the differential equations. Here they are, okay? I have to know, though, that the heights are coming in as y, and the height of tank 1 is y0, and the height of tank 2 is y1. and then I return as a list the two rates of change, right? So there's quite a bit to do there. And you can imagine that would be a lot to do if the model was big. But I'm not done yet because now I have to ask the computer to solve the equations. And this is what this bit does. I have to set up some initial conditions as a list. I have to create some time points using linspace. I then have to call the solver. Then I have to, and then I have to plot the results. Put in some labels, show a legend. So in all, we've got 1, 2, 3, 4, 5, 4, 5, 6, 7, 8, 9, at least 10 lines there. 11, 12, 13, 14, 15, 16, 17, 18, 19, at least 20 lines at least just to solve these two little differential equations, okay? Lots of opportunities for mistakes, all right? The other problem with this is the only person I can share this with is another Python user. If I wanted to share this with a MATLAB user or a Julia user, they would have to rewrite this. And that would be a bit tedious to do, right? Especially if the model's big. Okay. Anyway, if I do run it though, I get an answer. So I'm starting off with both tanks empty, and here we go. Time, follow the height. We see the water tanks filling up, right? Looks good, right? So we've got the simulation working. Now that's sort of hard way to do it. I'm going to show you a preview now of Tellurium, but Lucy will give you more of this, of course. This is one way to do it. I import Tellurium. I then specify my model. So these are the two differential equations. I set up my parameters. I ask it to do a simulation, and I ask it to plot it, and that's what I get, okay? I'm cheating here a bit. I didn't specify the labels, but I could do that in the plot. Now, the number of lines are shorter, of course. Maybe that's not a big deal. But the advantage here is I can share this with anybody because I can export this in SBML and give it to somebody who uses MATLAB, somebody uses Julia, somebody uses Java, somebody uses whatever language you want. And they can reproduce this model exactly, okay? So that's one advantage. Anyway, so you may be wondering what all this is. We'll come back to Lucian will say a little bit more of what all this stuff here means, okay? Now let us have a little quick look at biological models. Biological models are not water tanks, but they're pretty similar though in some respects to water tanks. Biological models, at least in cells, are made up of chemical reactions, or usually enzyme catalyzed chemical reactions, okay? And we have our own particular laws for describing how fast chemical reactions go. Here's one, for example. So if I have a chemical, substance called A gets transformed into a substance called B, the rate at which that happens is actually often just proportional to the concentration of A. And so is equal to some constant K1 times A. Sort of similar to the water tank model. We don't have a square root in there. In fact, it's simpler than the water tank model. The rate of reaction is simply directly proportional to A. What happens if the reaction goes both ways? Let's say it's reversible. Then I just put a minus sign and put the reverse term. And that's it. In fact, with just these two, you can build a lot of models in systems biology, okay? The only thing I haven't mentioned is what happens if I have a biomolecular model or a bi-model or whatever. I think I've got that coming up, I think. So our models are basically made up of lots of reactions like this with some kind of rate law, initial conditions, and then we run a simulation. Now, in general, what happens is with Tellurium, we would just give it the list of these things. And then from that, it'll construct the differential equations for us. Now, if you are writing in pure Python or MATLAB, or whatever, you'd have to write out the differential equations yourself. I mean, that's not difficult to do. And here's an example if I have, a little mini pathway here, which has a single, maybe this is a metabolite, S, that's made at a rate of V1 and then gets used up at a rate of V2, the differential equation that describes the rate of change of S would just be V1 minus V2, okay? It's again, the conservation of mass is very much like the tank model. You just sum up all the things that come in and subtract sum of all the things that leave, okay? What tellurium does is it constructs these equations for you automatically. In fact, most of the tools in this space, that's what they do, okay? Here's a bigger example. So this is in sort of two branches, one after the other. So we have a reaction coming in, it makes S1 gets consumed by V2, and S1 gets consumed by V3. But then the V3 enters S2, and there's another branch. And you can imagine, very large networks like this, lots and lots of reactions, lots and lots of molecular species. We can turn this one into a set of differential equations in the same way, very much like the tank model. So S1, we sum up all the things that are going in. There's only one thing going in, that's V1. And we sum up and subtract all the things that are leaving S1. So that's minus V2 minus V3. Okay. So you sum up all the things that go in, you subtract all the things that are leaving. Okay. Same goes for S2. This time V3 is coming in, and the two things that are leaving are V4 and V5. And you can do this no matter how big the network is. Now, there are some variants if you have different stoichiometries and so on, but essentially this is what it is. In fact, here's an example actually that has a stoichiometry, a different kind of stoichiometry. So this one, the first reaction in particular, we have A plus X gives 2X. All right. So somehow this reaction involves X transforming A into another X. This is why we get 2X. So the question is, how do we represent this as a model? Well, that's not so difficult. Most of them are the same as we did before. For example, the rate at which A is consumed is just minus V1. Nothing makes A, okay? So there's no A coming in. Something uses up A, and it uses up A at a rate of V1. So that's why I've got minus V1 here. Y is somewhat similar. Y gets made at a rate V3, but gets used up at a rate V2. So I got V3 minus V2. B is actually straightforward. B just gets made at a rate of V3. So I have just V3. And let me do Z at the end here. Z or Z, depending where you come from. Z gets made at a rate V2 and gets used up as a rate V3. Now, X is the odd one out here because in fact, I think this is wrong. Hold on, no, it's right. Yeah, it's right. So this needs a little bit of an explanation. The minus V2 is straightforward. X is being used up by V2, okay? Disappearing at a rate V2, therefore it's minus V2. The odd one out is the V1. And the V1 comes from the fact that A is being used up by V at a rate V1, but it's being made at a rate of twice V1. All right, so if I add minus V1 from this and add it to plus V2, I get plus V1. I don't have a do I have a pen here? Let me see. Can I do that? So it's actually minus V1, right? It's not a very good one. Sorry, yeah, minus V1 plus, it's getting made at twice. That's a terrible V2. Can I get rid of that? Yeah. Really hard with the mouse. Plus V1, right? So that's the rate at which X is changing. And so if I add minus V1 onto 2V1, I'm left with V1. And that's why I have V1 there. All right. So that's one thing that stoichiometry can do, okay? But lucky for you, tellurium handles all that. You don't have to worry about that. With tellurium, you literally, how do I get out of this? You literally just write these out, okay? That's all you do. And it'll do all this other stuff for you. So you don't normally have to worry about well, you never have to worry about the stoichiometry. But if you wondered how it happened, this is how the magic happens. It takes into account the stoichiometries on both sides and adjusts the rates accordingly, okay? All right. So with the water tank model, we saw there was a special law for how water left the tank. It was to do with the square root of the height. We have our own laws. We saw one here. This is called the law of mass action, okay, where the rate is simply proportional to the concentration of A or B. But there are a lot more sophisticated laws as well, and those of you who've done biochemistry will be familiar with these. So at the top, I have the mass action one, which is the simplest, but also we have the enzyme ones. So the Michaelis-Menten one. So this is the irreversible one. Those of you who know biochemistry will recognize the Vmax and the Km. More realistically, though, you'd use the reversible one, okay, which also includes the equilibrium constant and a reverse Km. And there are a lot of variants on this, depending on how many substrates and products you have and whether you have regulation or not. If you're working with genes or simple regulation, you might have a simple activation law like this. So it's what's called a Vmax times the activated to the power of something constant divided by a constant plus the activated to the power of a constant. Some of you will recognize this. This is essentially the Hill equation. Okay. If you want an inhibitory one, you just leave out the top term, and you end up with this, and that's the inhibitory. We'll be looking at these later. These are the kinds of rate laws you might use in a real model. And there are a lot of different kinds. And sometimes you can get away with just using mass action. Say for a signaling network, sometimes you can just use mass action. For metabolic network, you almost always have to use Michaelis-Menten, and you really need to use some kind of reversible form. If you're working with gene regulatory networks, you can get away with using these sort of Hill equations and so on, okay? So, but this will be down to your own knowledge on what is required, and there could be an entire summer school devoted to the kinds of equations you need to use in your model. But for now, we'll just use mass action and maybe some Michaelis-Menten, okay? One last thing, and I think, yeah, this is the last slide. Something we'll talk about quite a bit is this idea of a steady state. So we'll talk more about dynamics soon, but one thing I just want to mention at this point to plant it in your brains is this idea of a steady state. So the tank model, we go back to the tank model. Here's the tank model. Here it is. I just want the picture there. You can imagine that if you had this running, that the level of water would eventually reach some constant value, where the amount of water coming in is equal to the amount of water leaving. And a lot of systems do this. They sort of tend towards a constant state. And this is called the steady state. And when I did the simulation, although I didn't mention at the time, you notice they reach a plateau. Right? The heights of water reach a plateau. And we call that the steady state. Okay. And I would say all biological systems have a steady state. Sometimes you don't see them. We'll talk about that tomorrow. But most of them have them. And when you run one of these models, like, this branch model, you'll find that over time, S1 and S2 will reach some kind of steady state. If it doesn't reach a steady state, it'll either do something odd like oscillate, or it'll go to infinity, or worse still, go negative. When that happens, you know your model is bad. Oscillations are fine, but if your concentrations go to infinity or go negative, you know that's a problem, and we might look at a few instances of that later on. But anyway, I want you to just put in your mind this idea of a steady state. Most systems can approach a steady state. Anyway, so that's my slides. That's sort of brief introduction to modeling. I hope that will get you started. Do we have any questions coming in about that? Are we good? Okay, we got some answers and questions. Okay. All right. In that case, let's get started, shall we? Yeah. You ready then, Lucian?
Yeah, sure.
I'll stop sharing.
As if we're good
Okay.
at covering network motif modeling.